Yeah, I can play. I'll just leave my. I think she says see you tomorrow.
Hello, everyone. Thank you very much for coming through for this session, and must apologize for starting a bit late. I feel very important. I have my own mic, and the others can exchange. We have our 4 colleagues here, and a 5th colleague is supposed to be sitting here, who's Dr. Vera from Havana. Is not feeling too good, so I was just talking with him now. And I said, for whenever he feels a little better, can he sit here for 1 or 2 questions? So I'm saying this early on so that my colleagues and everybody don't feel disrespected. People come whenever they need to. He's just not feeling well. He's sitting outside at the moment, okay? And it's partly why we're starting late. We are in a program where we are going to look at vaccine capacity within South Africa. We want to look also at the VLP technology. This is virus-like particles. Those who are not scientists among us, it's more like, how do I make a thing that looks like a virus, but it's not as lethal as a virus is? I think, yes, he's nodding. Yes, so between our esteemed panel, I will be asking a few questions. Please, I'm not the one who knows about these things. It's more or less just to facilitate so that we know where we are. and I think we'll be here at the end. I will give a chance for some questions to direct it to our panelists, and we can see how far we can get with it. I've got Dr. Tseko, another of the CSR products, I can say, which is wonderful. Welcome, Dr. Tseko. And I've got Dr. David and Dr. Yeni. As far as I know, they are experts in the VLP technology. This is why I introduced them together. I've just met them recently, so I'm not going to say a lot about where they're from and all those, and I'll ask each one to introduce themselves as we go. And I've got Dr. Ntageni from the OBP, and all of them have knowledge and understanding into production, technology, and vaccines in whichever capacity, or at least biosimilars and antibody production. Now, I will ask each one to do so, but I'll start with Dr. Tseko, as he introduces himself, to say, in the South African space itself, and what he understands about vaccine biosimilars. I'm putting them together because my little science says the structure of production is very similar. So when I use them interchangeably, that would be the reason. What is the capacities, what is the infrastructure, also as far as the CSR is concerned, but also looking at the countrywide. Thanks, Dr. Tseko.
No, thank you, Pierre and Dr. Levere. And, Dr. Levere said I'm also a product of the CSR because he is one, and therefore a colleague of mine. Very well. And so thanks for the opportunity to be part of this panel. I come from the BioManufacturing part of the Council for Scientific and Industrial Research. I'll be brief in introducing the organization because I expect that many of us in the room have at least are aware of the CSR. We are a science council mandated from an act of parliament and a multidisciplinary organization that does a lot of things from defense, engineering, IT, but the part of the CSR I'm from is a division called Advanced Chemistry and Life Sciences, where I am part of the biomanufacturing impact area. So some of my colleagues are here from the biomanufacturing impact area, where really our main role, at least in the area that I am confined to, is to provide skills and infrastructure or a capability that can assist many different partners. And when I talk about this, we often talk about translating technologies from universities towards commercialization. We work in partnership with small and medium enterprises. We work in partnerships with also global partners to localize technologies. And I think that context is important here because my colleagues to the left from San Diego, they will introduce VLP Bio, are shortly going to become our partners in localizing some of their technologies. What we have at the CSIR, and I'll start there, and then expand, at least with my view of what is available in the country. At the CSR, as I say, we have an approach that we want to be open access, so that because we are a publicly funded institution, we want to be able to allow anyone. Of course, there are mechanisms and we do need funding support to make this happen, but we allow anyone with an interest in translating biomanufacturing technologies towards a commercial reality to access our resources. We have a fermentation capability right up to 1,000 liter scale. We have also a unique approach where we have utilized plants called Nicotiana by Tamiana to also make some of our biotherapeutics and vaccine candidates. We are currently now with excellent support from various international partners installing even mammalian-based CHO cell production, because we realize, and I think all of us in the whole world realized quite acutely, and we've said this many times over the past years, that the pandemic has taught us that despite the global world that we are part of, it's important for us to have regional, local capacity to do things that are important for sovereignty and self-sufficiency. And so in that context, we want to slot in the translational space where we work with others to prove concepts. And a major development that I will highlight in my introductory remarks is our facility that we're currently building right now, which will operate as a facility for producing phase 1 clinical grade material for investigational operation. So we call it a small scale investigational CDMO-like facility. It's a long mouthful, but the idea is that all of the developments we have advanced so far to preclinical stage would require that we hire and pay significant amounts of money to overseas service providers who will produce these batches. So one of the things we are building to plug what we see as a major hole in the national system right now is to develop this CDMO facility with support from government, support from some international philanthropies and European governments. So around the broader landscape in the country, I believe that a lot is happening. And I believe that what we are seeing right now is actually a bit of a revolution in terms of advancement of capability for South Africa. Because I'm aware, for example, Dr. Ntangeni and his colleagues, I believe, have just invited me to the launch of one of their facilities. I'm aware of developments of expansion at BioVac. I saw the African CEO is here for another session to talk about some of their developments there on mRNA vaccine production. The last comment I'll make is that I think it's very, very good that we have all of these developments happening, because I think that it's important to also have, as a country, some focus, but it's important as Africa and as a global world to have a diversity of platforms that we rely on. Because what we saw with bottlenecks regionally and globally around single platforms is that it can actually compromise our ability to respond to the situation we saw a couple of years ago. I think for introductory remarks that was sufficient.
I concur. Can I do this, Dr. David? I want to give this to Dr. Ntangeni, and the reason for this is that I want to just deal with the capacities so that we're able to come back and say, if these are the capacities, this is what we have in the country. How do we see this integration with the VLP, so that for my audience, there's at least a follow-up on that. I'll probably ask Dr. Vera to, after Dr. Ntangeni, to just tell us, he is from Farm in Cuba. This is a well-known area of biotech, but also in the collaboration model, because one of the things we do, I know because I was there too, it's very important that the technology is seen as me, and not as that is the objective we need to do. This is my group, this is me who's running this team, and if I'm not visible, maybe the technology must die. So I want just a small, also you can tell us what they do at Farm, what is the collaboration, but obviously more along the animal vaccine and what they do and how they're able to collaborate. His English might not be as 100% as you're used to it, but he's very good. I've been talking with him. So that's why I'm posing his question early, so that while Dr. Ntangeni is talking, he can synthesize it. Over to you, Dr. Ntangeni, in terms of your introduction and also knowing you are with the Honest of World Bioproducts, what technologies, what are the capacities and all that, and also knowing your side of story. I know you've been in the field. I will tell you a small story. This is one man that I know can deal with a very stubborn microorganism and get it right. I used to wonder all the time why and how does he do that. So over to you, sir.
Thanks very much. Like the facilitator has indicated, I'm Dr. Ntangeni. I represent an organization or a company called Honest World Biological Products. In short, they call it OBP. Probably most of you have not heard of it, but I would like to say with assurance that it has impacted all of you, especially if you are not a vegetarian. The chances are that every day when you consume meat, whether it is mutton, goat meat, beef, that meat comes from an animal that has been vaccinated by a vaccine from OBP. And there's no way in the lifetime of that animal that that animal was not exposed to a vaccine from OBP. That is how big OBP is in terms of its impact. And in as much as it's a state-owned entity, it operates as a commercial entity. It does not get subsidy from government as such. It relies on its revenue. And yeah, OBP, in as much as it is a national company, we are also global. We are global in the sense that when there are shortages of vaccine in the country, OBP gets accused that it has prioritized its global market. So that is the global reach that OBP has. And in preparation for its global spread, we have even invested in the new facility which is called the current GMP facility, so that we can produce our vaccines in accordance to international standards. So that is the bigger picture in terms of OBP, where it is going. OBP, in terms of vaccines, we produce over 20 million doses of vaccines in a year. And those vaccines, they are vaccines based on stubborn microorganisms or viruses or even parasites. We do have a number of vaccines. And the technologies which have been using for years and maybe for centuries, they have been based on inactivated vaccines. Others are live, attenuated. But we had to move with time. And as a result of that, there are new technologies which we are adopting. We are not necessarily developing new technologies. We work with various establishments. CSIR is one of them, and universities where we identify technologies of interest. And our main field of competence is in the field of scaling up production processes. Very often universities, they tell us that they've got this very fascinating candidate to be a vaccine, and when we tell them what it needs to take to transition that candidate vaccine into a vaccine, and we tell them to take up to 10 years, somehow they don't believe us, but when we start to get to do it, to execute it, then they realize that that is what we've correct what we've been saying. But very much important, in our scaling up, we have got production processes which go up to 1,000 liters. So when you bring that candidate vaccine in a 5 ml tube, the process that we embark on is to scale it up to manufacturing volumes. And apart from that, we do clinical trials, and these clinical trials, we use them to test if the vaccine is efficacious, if the vaccine is safe, and if it can be registered. And we do that following international clinical protocols. So that at the end of the day, when you get the vaccine, you know that this vaccine indeed it works. And as a result of that, it is not that we only invested in the production capacity, we have also invested in small animal facilities. These facilities where we produce, we've got more than 20,000 small animals. We then call them rats or mice. We do have the hamsters, guinea pigs, rabbits. So probably we host the largest colony of all those small animals, laboratory animals, which are used in the development of vaccines. And what has been a drawback in the country in terms of vaccine development, it has been the availability of a facility which is vector proof. When we say vector proof, we mean that in that facility you can house animals, and as the animals are housed, they are in an environmentally controlled facility. The temperature, the humidity, and the number of particles in the environment. And apart from that, even a mosquito cannot get in. And yes, even a mosquito. And if you have to get in there, you have to strip, you have to wash, and then you put proper gowns, and then you go and work with the animals. So that facility is one of its kind in the continent, which is we are going to be launching in the very, very near future, probably in terms of weeks. So that is the capacity and the capability of OBP, not to mention human resources in terms of scientists that work there, PhDs with a lot of experience. And apart from that, we train and graduate on average about 3 PhDs or postdocs in a year, and still as a state-owned entity. Thank you.
Wonderful, thanks, Dr. Ntangeni. Dr. Vera, if you can just briefly give us some of the experiences at BioManufacturing.
Yes, as Dr. Filani told you, you will excuse my English. It is not very good. But we are from Cuba, from the company named BioManufacturing. BioManufacturing has been producing animal vaccines for a long time now. We started the industry in 1960. It is more than 60 years ago. We have developed a range of vaccines for all the animal species that we have in Cuba. But maybe our range of vaccine is not very wide, because we can deal only with the microorganisms that are present in Cuba. We have not foot and mouth disease in Cuba. We have not rinderpest. We have not avian influenza. Then our range of vaccines are the vaccines that we use in our livestock there. We have more than 14 viral vaccines. We have more than 15 or so bacterial vaccines, and also we produce diagnostic kits for the diseases that circulate into our country. Now we have a deal, a partnership with Tocavio, with Dr. Filani. We will try to help to produce some of the vaccines, mainly avian vaccines, here in South Africa. We will try to get our best knowledge and our best help to have these vaccines quickly and with good quality. It is our goal. I think we can reach it, and let's see how in the coming time we will be able to present you some results of the vaccines here in Sudan. I don't know if you have any questions. Now it is not necessary for me to tell a lot more of our company. It's the main idea.
Thank you, Dr. Vera. And I just explained before you came as well that the collaborative model of the Cuban industry, in this case in biotechnology, is really important in terms of what we can learn as a country to maximize our resources to get what we think we need to. And for now, I will invite both Dr. Yeni and Dr. David to say, having looked, first introduced, but also just a small introduction of what VLPs are. I know I butchered it, but a bit of what they are, and also if one looks at the South African context, having just looked at the two also available capacities, what are the synergies, what is it that we can work together, and how do we pull from here? And after that, given our time, and please, if I keep looking at my phone, there's no messages, it's the time I'm trying to look at, no disrespect. So, so that we can wrap up to see what would be the way forward with the kind of work that is available, and what do we do. Of course, we cannot finish it today, but a very good introduction to say what are we looking at and what is the way forward. Thank you.
Thank you. So my name is David Whitaker. I'm a scientist at VLP Bio in San Diego. And so we work on virus-like particles. And so the advantage of these is that the immune system has, actually I might take a moment just to thank Africa Bio for inviting us here, because it's been very exciting actually to see. I think there's a lot of energy, and it seems like the right time to get involved as an innovative company in what's going on in South Africa. So we learned that, and so it's very exciting to be here. So the thing that we work on is the VLPs, we do think is a good match. The virus-like particle has been recognized by the immune system of animals as something dangerous naturally. And so what our technology is to take this VLP and you transfer epitopes, so pieces of other foreign proteins that antibodies are made to. And those antibodies will block infection or the toxicity of viral or bacterial toxins. And they transfer that mutagenicity of the particle to this other target that you know of. And there's many of these targets out there. And because the VLP itself is not infectious or dangerous, it's very easy to manufacture. You don't need to worry about infecting your workers. It's very stable. We can dry it, or we can just keep it at 4 degrees. It doesn't need to be frozen. It's produced, and I'll let Yemi talk more about the production, because that's his expertise, but the production is easy. And we have our targets that we've worked on for years, and we always strive for a low cost of goods being built in or growing bacteria, and then also having a platform to give us some flexibility. So there's a great deal of information out there in the scientific literature of what antibody targets will produce what you want. So protection from viruses or bacteria or other things. And we can use those to put on our platform and then create a vaccine that is deployable. I don't want, at the risk of going too long, maybe I should pass it over and let us have a question and answer about what's
Thank you very much for that.
Okay, well, thanks everybody, and thanks for the invites to actually come out here. We are VLP Biotech, and as David has introduced what VLPs are, I should make a contrast about the types of VLPs we're working on. Because we know VLPs have been in the mainstream now of vaccine technology for probably the last 30 or 40 years. Yet, some of the key limitations of existing VLPs are manufacturing, and also not being able to actually have a broad platform to go after different types of diseases. Our platform is a chimeric VLP platform, which means that you can actually engineer this platform to display epitopes from foreign pathogens. And as a result of that, it means you can actually, based on this platform, create a pipeline of vaccines that will be readily addressable to different types of diseases. So in fact, our founder, Dr. David Willage, is one of the world-leading experts in hepatitis B immunology. And the earlier work of our platform was to engineer, to fully characterize these hepatitis B viruses, to understand what parts of this platform or these viruses or virus-like particles are amenable to being engineered to display those epitopes. So how we got involved with discussions today and why it's highly relevant today. really is the understanding, especially as we saw with the recent COVID pandemic, which was that if you look at global capacity for production of vaccines, of proteins generally, upon which many of these vaccines are based, there's definitely a concentration in, I guess, in Western countries, as well as in Asia. But even that capacity is not sufficient for the 7 billion people that we have in the world. So there's a reprioritization that you find, which is driven by national borders and politics. And it's very important to be able to have self-sufficiency when it comes to existential health security products like vaccines. So we are deploying this platform for that purpose. The purpose here is to be able to provide a platform that would enable for local production, not just for human vaccines, but also for animal vaccines. And that's compatible with existing manufacturing infrastructure. So what do I mean by that? Well, it's very great to learn from Dr. Seppo and Dr. to my left, all the capacities that already exist in South Africa for vaccine production. And also to learn that our platform actually is expressed using E. coli, which I think is one of the few VLP platforms globally that can be expressed in this expression system. Importantly, they are highly stable platforms. In fact, one of our manufacturing processes is to heat up our, I guess, supernatants or lysates up to 60 degrees, where everything else falls out of solution, and you have a stable VLP, which we've actually assessed for over 3 years and shown it's still stable. So that's important for the cold chain, because as I said, we want a pan-African vaccine platform. So it's a very versatile platform. I think it's one that will be relatively straightforward to transfer the technology and know-how in order to be able to get up and running very rapidly. And I should also say one more thing about VLPs. Traditionally, vaccines have relied on, as has been said here today, whole organisms or inactivated organisms, but all of these always have the chance of being reactivated, and so they are quite pathogenic in some respects. So really being able to rely on a VLP that is not capable of reverting back to being a virus is very important. So we actually have what we consider in this case to be very safe vaccines that we want to deploy to the African continent. So I'll stop there so that we can have time for questions. But we're, again, very happy to be here to have this discussion.
Thanks, Dr. Yeni, for that incisive talk. As you will see, especially being from the scientific background first and try and do business, I myself am guilty of turning a lot more towards the science because I'm comfortable talking about it. And it is welcome and it's wonderful. What I'm trying to say is that at the end of this, please feel free to also ask questions when you meet them rather than only at this. I'm going to open for questions, burning ones. I have a few for each one of them so that I can close, but I want to also give a bit of time for our audience to have some questions. I'm going to run the mic.
Yes.
I'll take 2 or 3 at the same time, and then I know we can get there.
Good day, my name is Gita, and I'd just like to ask, in terms of the manufacturing capacity, during COVID, I think there was different vaccines available, but due to the different variants or strains, like Beta or Omicron, there was a battle because I think most of the vaccines that were manufactured, they used the Alpha strain, of which it couldn't fight other different strains such as the Omicron, which were much more stronger. And so I think, what are the interventions put in place with that? Because as you mentioned, I think you do different vaccines, the chimney-toad one, the live ones. So what are the interventions put in place for that in case of now we know that maybe other pandemics or epidemics could come in place within Africa?
I'm going to ask Dr. Yeni and Dr. David, and I'll rephrase the question to cover multi-strains with your VLP technology. What kind of interventions are there to do that, if you can do that? Sorry, I'm going to take three. I'm just allocating the questions so that we can go fast.
I just wanted to know whether to start shouting. Good morning, everyone. Thank you so much for such a wonderful talk and presentation about the vaccine and the new strategies and the approach that has been taken to make it much more cost-effective and bioavailable to the entire population after COVID. That is what the whole world is now looking after. I want to gain more information and understanding about the VLP, that it mimics, like we are using E. coli, a prokaryote, to create a eukaryotic product. So when it comes to designing and development in a prokaryote, a eukaryotic protein, are there any difficulties that we encounter when it comes to folding and the processing of that epitope, so that it becomes much more effective and bioavailable to the antigen presenting cells, so that it can mount in not only an immune response to the present epitope, but it also is able to create a memory response for prolonged memory, for a prolonged time. Because that's what we achieve in vaccine, that it has to be a prolonged immune response that is being taken up by even the other APCs. So that is one of the understanding that if you could just share some light on that, that would be really helpful.
I'm not going to rephrase that. You will share between the two of you. Another question? Good. I think what I'll do as well, if you allow me, I'll ask Dr. Tsepo so that by the time these questions are asked, I'll ask the other two. The question here that I wanted to ask, especially as we're looking at the, yes, I work with Dr. Tsepo, so I call him Tsepo, so every time I have to think, is Dr. Who? So here I want to know from the CSR's perspective, and when I say CSR, I'm really talking in the South African perspective, which kind of platform, which kind of targets are we looking at, and what are we looking at in terms of me having an understanding of what capacity are we trying to build first up as we develop, understanding this is still a development and not necessarily putting a final product on the ground. Thank you.
So I just have to make sure I don't get too down into the weeds. But the first question about the variants and kind of keeping up with the evolution of the pathogen. And so our technology is epitope focused. And so what that means is your typical virus, vaccine, and this would happen with spike protein for COVID, you put the whole spike protein in as a vaccine, the immune system makes the antibody it makes, and it goes to the immune dominant regions. And those are the ones that then receive immune pressure. So again, if I have too much jargon, I apologize. But that causes the viruses that mutate away from the viral or the antibody neutralizing response. It forces that evolution. So our strategy for COVID was actually, because we have epitope focus, is to go for epitopes on the spike protein that are much more stable, that are required, so that mutations within that region will disrupt the function of that protein. And because we have epitope focus, we can do that. I don't think any technologies have really gone out after that. There's some research papers out there. We did present to a group in the US, BARDA, and they liked the technology, but they were looking at the time for people much closer to clinic. So we're kind of, at this point, trying to build that in. So there is that answer. We can also, because it's bacteria, we can iterate very fast. We could, in principle, follow the mutations. The limitation for our technology and mRNA and everything else is, by the time you've gone through the regulatory process to get that vaccine approved, the next variant is up. And so that's why we like the idea of going after conserved epitopes. This is true also, this will work for like flu and a lot of the other epitopes out there, a lot of the other pathogens out there. And then the question about the technology, I have to be very careful not to get too down in the weeds. So it's based on the hepatitis B virus, nuclear capsid. So that virus has a surface protein, there's an envelope around it, and that's what is involved in infection of the human liver. We don't use that. Inside that is the nuclear capsid. It's a single protein, unmodified. There's does not require phosphorylation. It does not require any post-translational modifications. So their bacteria is good, because we don't want that. There's no glycosylation. And so that particle, so in a natural infection, Hep B, you get infected once, you'll make antibody to that for the rest of your life. That is the clinical test for having been infected by hepatitis B virus. You are anticore positive. That's why we chose that particle. And so when we construct that protein, the single subunit protein, with maybe a conserved COVID epitope on the tip of the spike, where most of the antibody is made, we make it in bacteria, it self-assembles into a spiky ball, and the idea is that antibody gets made to the end of those spikes. And then we don't know, we have not been to humans, so we don't know our chimeric VLPs that make antibody to COVID or whatever it is, malaria or anything. We haven't been to humans, so we don't know the longevity. So that is something we're working on. Getting really down in the weeds, we can talk later about T helper epitopes and specific things. We have strategies for that, but we're basically excited to learn how that'll work. And I think those were the questions, if I'm correct.
So, Pierre, if I recall your question correctly, it was around what we are looking to apply this platform on. And I think we're now free to talk openly because yesterday many of us are aware that some of our seniors in government announced the inception of the vaccine innovation and manufacturing strategy, which by the way, weeks ago, we were introduced to VLP Bio in that context. And the exciting parts of that the CSR will be involved in, for example, are around developing vaccines for HPV, for example. And then we know the context there is that cervical cancer is still a hugely burdensome disease in South Africa, and South Africa suffer from cervical cancer that develop from HPV infections. And in our context, in the public sector, the vaccine available currently does protect against 70% of the strains, while those of us who have medical aids can actually access a more multi-valent vaccine. In this context, and I think our colleagues have already alluded to this, you can imagine a situation where using this platform, either through chimeric strategies or co-formulation strategies, or more importantly with the epitope-focused approach, selecting very specific regions of these viruses that would give us this broad specificity would be quite powerful. So you can see that this approach for that particular quite pertinent problem to our society is something that is poised to make a big impact. I'm also quite keen on the idea that through a platform approach and having worked on manufacturability and having worked on even downstream processing of these products that we make, we will be able then to respond much more readily to recurring other diseases simply because the host cell protein profile, the actual physical characteristics of the bulk of the material we produce will be quite the same, despite the epitopes being different. For me, that's very powerful and exciting to think about, because then we can apply this quite more broadly to a number of other opportunities. I know that in the broader strategy, in the broader community of collaborators, there are many other targets that I'll be quiet to talk about because other people can talk more openly there. But HPV is one that we're very much interested in.
Thank you very much. I think because you covered the two questions, I'm going to go just for the last two questions. I think lunch is very important. Dr. Ntangeni, I know given the batches and batches that you produce every year and this new facility that you are looking at, and I'm very thankful, and this is very exciting by the way, Dr. Sekwa, in that for the first time, if you ask me, there's a concerted effort to be able to produce that which we want, not buy. We don't have the money, just to produce it, work on it. It's not only to help the people that are the end users, but also develop those technologies and indigenize them and be able to do the things we need to. So it's very exciting. And yes, this is my excited face. It doesn't look very different. So it's a cold water and hot water. They do look the same. Dr. Tangeni, I wanted to say that with that and also looking into the continent and not only just South Africa, I know the mandate is that of securing food security in the country. But with that kind of intervention, with that kind of infrastructure, when you look into the continent as well, how do you see such infrastructure assisting in terms of the livestock and everything within the continent? And how do we as South Africa contribute towards the development of livestock?
Thanks, Facilitator. I think what is important is to indicate that OBP, in as much as historically we have been so much focused on inactivated or attenuated vaccines, we have been progressing with time in terms of technology. And the VLP technology is not necessarily new as it comes to OBP. We have gone to the extent of even having products which were patented, which are based on VLP. We have developed and even published VLP-based product for blue tongue vaccine and also for African horse sickness vaccine, together with some of our collaborators, UCT and also the CSIR. Where OBP comes in there is to provide the antigenic epitopes, which are important in terms of being vaccine candidates. And what is important is that we have got various serotypes. When we say it's a blue tongue vaccine or it's an African horse sickness vaccine, you may find that we've got 2 bottles or 9 serotypes. So it is not that we would want to make all those 9 serotypes the way that we do, because you produce each vaccine, each serotype one at a time. But if you can have a platform where you can even put multiple epitopes in one construct, whether it is a chimeric VLP, that would be something which is very much appreciated, because it brings down the cost of production. And more importantly, there's always this issue that if you are having live attenuated vaccines, there are chances of reversion to virulence. So with VLP, you nullify that. So there's no chance that the attenuated vaccine or this vaccine will revert back to virulence. So it becomes very much important. And we've gone to a point where we, in our facilities, where we tested the clinical efficacy. And we have developed models which we can use to test the efficacy of the vaccines. VLP biotech, they can have the technology, but they may not have the facility to test if it works. So that is where we come in. And the diseases which are relevant, that is where we come in. And the market which is relevant, and how our distribution network, that is where we come in. And maybe the approach to this should be multifaceted, especially if you consider the African continent. And because the issues sometimes become like, no, we don't need GMOs in our country. So when we put a vaccine and you want to distribute to certain parts of the continent, then they will say, no, we don't deal with GMOs. So there needs to be public awareness in terms of the equivalence, in terms of GMO, in terms of whether GMO can impact the animals or not. So that is where the challenge is now. But in terms of the facilities, in terms of the processes, all that we wanted at some stage is to have a facility where we can have, because the expression systems which we were using was based on tobacco plants. And some people, they don't want to smoke tobacco while they're eating their animals. So it was to find a way to process the materials, whether to plant in a big space the tobacco plants, and then to have a processing facilities which is acceptable in terms of GMP standards. But also it is very much encouraging if we have got a bacterial system. It is easy to grow, it is easy to scale up, it is easy to do downscaling processes. So that would be very much appreciated. Thanks.
Thank you, Dr. Chage. If there's one burning question, I'll allow it. Otherwise, I'm going to wrap up with Dr. Vera, and we move forward. Any question? Okay, I'm going to ask that maybe the questions don't have a commentary, like we just shoot to the question.
Thank you to the panel for the information. I'll be straight to the point. My question is specified to the VLP team. Are there any products that have been licensed using this technology? I'm just trying to assess if regulators have gone through this and it will be easier for the upcoming product. Thank you.
Thank you for the presentation as well. Just to be quick and brief as well. Extending from the question that I was just asked, I wanted to also find out, maybe also adding on what OBP has just said. Have you tried the platform using multiple viruses in the production system, the E. coli based one?
Thank you for the brief questions. Can I ask my panelists to please?
Well, I can start with the response in more detail. First question is a simple one. No, we haven't had any licensed products yet based on this platform. Yet, scientifically, we don't see any reason why we would not get licensure. The reason we haven't had licensure to date has been that in the original conception of this platform in the US, vaccines haven't been sexy. They've only started to get sexy now. Sorry for using that word. But there's strong interest in vaccines now. And we have candidates that are ready to go to the clinic to demonstrate clinical proof of concept. As far as the second question, which is multiple epitopes, I'll let David actually answer that question because there's really a lot of capability in terms of how many epitopes we can accommodate.
So that is a good question. I mean, they're all good questions. I won't get too down into the weeds. If you want more detail, you can find me afterwards. But yeah, so we routinely want to incorporate multiple epitopes, and we'll take both. We've not tried different viruses on a single VLP. Usually we're like for hepatitis B virus, we have a program, and we have about 8 epitopes that we consolidated down to 3 different regions, and we try putting them on one VLP. Also, when you want the most antibody you can get, the most immunogenic, most potent thing you can get, we found that putting it on 2 or 3 different VLPs has worked, but we can then manufacture in the lab. So grow them in the lab and lab scale. We can combine those and put those in animals, and we get as much antibody to each of them as if we get just put one VLP into each of 3. So we're very confident that I mean, there's an upper limit to how much we can put in one vial or on one vaccine, but we can do multiples. And in the research, as part of the research, we're developing that further.
Maybe I can just wrap that up, because the question about multiple epitopes is very significant. Because many times when you have a vaccine, the protection principle may be down to 1 or 2 specific antibodies. What we can do with this is really direct the immune response to raise those specific antibodies. So if it's a cocktail of those, then once we know what those epitopes are, we can put it on the platform. And we've really characterized so many sites in the VLP that are amenable to that.
Thank you very much. I'll have the last word. Dr. Vera, just the last one. I know in your country you have many institutes that contribute towards the same. I just wanted you to give us some of the collaborations that, say, for instance, Labio Farm would have if it were to develop something and what it would work on. And the reason I'm asking that is because while we do it back here at home, it's not always the case that we collaborate so effectively, and we wanted to get some lessons from that.
Well, let's say that specifically in Havana, we have what we call the scientific pole. This scientific pole agglutinates all the scientific, all the researchers, institutes, all the universities, all the scientific institutions of the country. And from there, we get a lot of ideas, a lot of innovations by research that are coming to our institution to adapt it to the big technologies and produce it for the fields. We have a very collaborative system in all the scientific institutions. If we are creating a new molecule, a new particle, we can, if we are not able to produce it, to have it, to introduce it in the market, we have to give it to whom is capable of doing it. And in the veterinary field, that is our field, we have received microorganisms from many other institutions. That they have to give us those microorganisms, not only for vaccines, only for bioproducts to control agricultural pests, only for other bioplaguicides. We have a lot of other biological products in our productions, not only vaccines. And we have received from other institutions many of their investigations during a lot of years. At the end, they have to give it to us and we will produce it.
Thank you very much, Dr. Vera. And thanks to everybody for attending. And I think this is only the beginning. There's a lot of lessons that can be taken from here, from the collaboration, from the new technologies, from the capacities, and how do we develop going forward as a country, and also as a leader in the continent, to make sure that the things that we have here can be replicated and help the whole country and the continent as well. Thank you very much, and thank you for your patience. I think lunch should be open now. Thank you.